heredity and evolution in organisms in different species. What is genetics? It's a branch of science or study of heredity and variation. So what is heredity? It's a transmission or a transfer of characteristics or attributes from parents, mother and father, to the next generation, that is offsprings. So the difference in character of the parents and offsprings, because there will be some difference in the characters, and this will be known as variation. So there are two types of variation. One is somatic variation, the other is gametic variation. This somatic variation, it occurs in the somatic cell of the body, while the gametic variation occur in the germ cells of the body. So somatic cell, they are not inherited and they are transferred or transmitted to the next generation. So they are known as acquired traits. While the gametic variation, they are inherited in the next generation. So they are also known as inherited traits. What are the importance of variations? First, these are the basis, or it is the basis of the heredity, and all the evolution, it is the basis of evolution itself. And the survival increases, this variation increases the chances of survival of the organisms according to the changing environment. So what are the causes of this variation? It can be mutation, recombination, random mating. So these are the most common causes of variation: mutation, recombination, and random mating. The recombination or crossing over is the important reason for these variation. It is basically the exchange of chromosome segment at the time of the formation of the gamete. Mendel and his contribution in genetics. G.J. Mendel, he was a famous mathematician. He was not a biologist. So he started his work on pea, garden pea. The name is Pisum sativum. So he worked on pea, and because of his excellent work, he showed what genetic is. He showed with certain observation and experiment, and that too mathematically. He was known as a father of genetics when he did that for 6 years, and he published the paper in the local newspapers and the local journals. He was not regarded, or you can say his work was not attributed. But 3 famous scientists, again from different corners of Europe, they found his work, and then he was regarded as the father of genetics. He chose 7 pairs of contrasting character. He chose P and 7 pair of contrasting character. See, this is a P, this is a gray and round P, this is a white and wrinkled P. There are 2 types of flower, violet and white. The different types of, say, this is a yellow pea, green pea. So he took 1 to 7 different pairs of contrasting character, and then he reached to certain laws, certain conclusions which are valid today, and he is the father of genetics. Now the main reason why he chose garden pea for experiment, that the pea has very short lifespan, around 2 to 4 weeks. The seeds produced are quite in a large number, so large number of seeds are produced. Then the self-pollination, the cross as well as self-pollination capability of pea, that was the reason he chose it. And also there are various contrasting characters that can be found in garden pea. So what was his law? There were 3 main laws: law of dominance, law of segregation, and law of independent assortment. What is the law of dominance? If the 2 alleles, what is this alleles? Alleles are basically the genetic content. We are talking about genetic content in organisms with a living plants, flora, fauna. So if 2 alleles at a locus offer, they differ, then one, that is the dominant allele. There are two allele, dominant and the recessive. So if the 2 alleles differ, then one with dominant allele, it determines the organism's appearance. Who determines? Dominant allele. The other, which is the recessive allele, has no noticeable effect on the organism appearance. This is the law of dominance. The law of segregation says if the two alleles, that is the genetic content for heritable characters, separate during gamete formation and end up in different gametes, so they segregate and they end up in different gametes. This is law of segregation. Then the law of independent assortment says that each pair of these alleles segregates independently of other pair of alleles during gamete formation. Monohybrid crossing. So one pair of, this is a pair, so one pair of contrasting characters, this was taken to cross two pea plants and it was known as the monohybrid cross. So if you see this figure closely, this is showing that the monohybrid cross between a true breeding, this one is yellow pod and this is true breeding green pod. So all the pods obtained was green in color. That is, when they are being combined, he found that all were green. This was capital G, the combination was capital G, small g. So the content was there from both these, but it was green. And the offspring obtained was known as F1 progeny. In the heterozygous condition, this is a pistil, this is a pollen. So when they are combined, or that they are supposed to be heterozygous condition, the parents are heterozygous here. So the phenotypically, what is phenotypically? Phenotypically is outside observable characteristic, while the genotypically is the inside genetic level characteristic. So if you see here, the parent are heterozygous, these two are heterozygous. So phenotypically, three purple flower were formed and one white flower was produced. But inside that, if you go genetically or genotypically, one homozygous, this one, two heterozygous, these two, and one homozygous recessive. This is dominant, homozygous dominant, this is homozygous recessive, and these two are heterozygous. All the three are homozygous. This is homozygous dominant, capital B, small b. Dihybrid cross. When two pairs of contrasting characters are taken to cross two plants, it is known as dihybrid cross. See, this is yellow round P and this is somewhat green wrinkled P. And these are the combinations. Say YR, this is small y, small r, and the combination at F1 was yellow round. It was this only. And when it self-fertilized, it can be any combination. So there are four possibilities here. These are four possibilities combination. So 4 into 4, it gives you 16 combination. These are all the 16 combination. So phenotypically means if you see outside observable characteristic, the ratio was 9:3:3:1. What is 9? 9 are round yellow. These are round yellow. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 round yellow. Then there are 3 round green. 1, 2, 3. 3 round green. Then 3 are wrinkled yellow. 1, 2, 3. 3 wrinkled yellow. And 1 was wrinkled green. But the genotypic ratio was somewhat like this: 1:2:1:2:4:2, 1:2:1, like this. Sex determination. This is used to define the sex of the child or offspring or next generation. There are certain environmental and genetic factors that is going to determine the sex, male, female, of the offspring. There are examples where turtles, see the gender of turtle is according to the temperature. So the environmental factor like the temperature also plays role. So what are the types of sex determination? There are different types of sex determination. XX, XY type, that these are found in humans. XXXO types found in insects. ZW, ZZ type in chicken, and in moth, butterflies, ZO and ZZ type. So the genetic factor, they include presence of sex chromosomes. For instance, in human, the presence of 2 X chromosome leads to female, while if it has 2 X, then it is female. If one is X, one is Y, it is male. Presence of 1 X and 1 Y chromosome forms a male offspring. In fact, in human beings, there are 23 pair of chromosomes. Out of these, 22 chromosomes are known as autosomes, whereas this 23rd, the one remaining pair of chromosome, is known as the sex chromosomes, or you can call it as allosomes. So sex of the offspring is determined by whom? By the chromosome inherited by the father, because father has this Y, mother doesn't have. Evolution. The sequence of gradual change. Gradual means the change is taking in a law, this time span is huge, that takes place in organism over millions of years, huge time, and lead to the formation of new species, is known as evolution. Lamarck, J.B. Lamarck, he was the first scientist who gave the theory on evolution. He gave the theory of inheritance of the acquired characters, and then Charles Darwin came. Darwin said, and he proposed the theory of natural selection or Darwinism. According to Darwin or his theory, evolution occurs through natural selection. So what does this theory of Lamarckism says? This theory also, we know it by theory of inheritance of acquired characters. It has certain postulates. So Lamarckism is based on certain postulate that the living organism, it will increase in size. It tends to increase in size in evolution when they evolve, and new needs lead to formation of new organs. It is not that the organ will remain same. New needs will give rise to new organs, and continued use of particular organ will make it more developed, more stout, more usable. And if you don't use it, disuse of organ leads to degeneration. New characters are acquired by individuals during their lifetime. Again, this is a gradual process. Theory of Darwin. So Darwin theory, also known as theory of natural selection. So what were the postulates of the Darwin theory? First is speciation or forming of species, so formation of species. And this useful variation, this says that useful variation from generation to generation give rise to the formation of new species. There is always struggle for existence because the food, space and everything in the world or the globe is limited, and due to multiplication of organisms and limited content and resource, there will always exist competition among the organisms. So only the best will survive, and which is the survival of the fittest or the natural selection. This says that nature would select those characteristics or those organisms that are useful or they are best adapted to the conditions, prevailing condition, and these conditions may be hostile. Speciation. What is this speciation? The origin of new species, coming up of new species, or evolution of the new species from already existing species is known as speciation. And speciation can take place through various processes. One is gene or genetic flow. This can lead to speciation, and this is basically a transfer of genetic variation from one population to another. There is random change in allele frequency, which is known as genetic drift, and this is also leading to speciation. Then natural selection, this is also a reason through which the speciation can take place. There are certain geography also involved, like the mountains, the river, the sea. So geographical barriers like these can also lead to speciation, and this is known as geographical isolation. Evolution and classification, these are very much linked to each other. Evolution means, development of different species from the previous species, and classification is devising or dividing or categorization of these species. So there are different evidences of evolution. If you see homologous organs, these are homologous organs. All these are nothing but hand or limb or forearm or forelimb. So these are the organs which are evolved from the same ancestor. Homo means same. So they are evolving from same ancestor, but they have different function. If you see, human use it in different way, as does the cat in different terms, while the whale is using this hand in different terms, like to swim, and the bat is doing it for different work. So the forelimb of horse or wings of bat, the flipper of whale, human hand, these are all examples of homologous organs, because they look same, but they have same ancestor, but they have different functions. Analogous organs, these are organs which have come from different ancestor, but they have same function. The function of bird wing, the function of bat wing, they have same function. Paleontological, or you say fossil evidence, this was also given for evolution, or this can also be used for evolution. The dead remains of organisms, these are known as fossils. The archaeopteryx, this possess feature of both reptiles and birds, and here we conclude that the reptiles has actually given rise to birds. So birds evolved from reptiles. There are two methods of finding the age of fossils. One is carbon dating, C14 dating, or by digging. If you dig more, or the thing which comes up first, it might have died. You can say died not later than if you go deeper. That means if you dig here, those you find here, they have died early, so they are not older. But here, those remnants are more older. So in digging method, the deeper the fossil is, the older it is. We have biogenetic law. This also states that the stages of development of an animal embryo are same as adult animal stages. Then vestigial organs, these are rudimentary in nature, and they have lost their function through evolution, like the appendix in human, the muscles of ear, and the wisdom tooth. All these are gone now. Evolution by stages. Evolution can take in different stages. So for example, evolution of eyes. The flatworms have rudimentary eyes. Insects possess compound eyes. Humans have binocular vision. So evolution of feathers is also an example of evolution of stages. Dinosaurs, they have feathers, but they couldn't fly. But now the birds are flying. They have feathers. Molecular phylogeny. So this is also an evidence of evolution. According to this molecular phylogeny, the change in the DNA during reproduction are the basic events of evolution. So organisms which are related to each other more distantly, they are supposed to, or they will have a greater difference in their DNA. Evolution by artificial selection. The artificial selection selects special phenotypic characters to produce organisms with enhanced characteristics. For instance, the plant nowadays, they are insect and disease resistant. The artificial selection can be used to produce different cabbage varieties. You say broccoli, cauliflower, red cabbage, all are the variation of cabbage only. Human evolution. Excavating, fossils, time dating, and the determination of the DNA sequence, These are all employed and used to study the human evolution or human evolutionary relationship. So the vast study of human evolution, it indicates that all of us belong to a single species which was evolved in Africa, and then it spread across the world in different stages. So what are these stages? The stages of human development are like this: Australopithecus, height around 1.1, weight 4, and he walked upright this many years ago, 35 lakh years ago. Then Homo habilis, height around 1.59, weight 50 kg. They started using some tool. They could speak 23 lakh year ago. Homo erectus, the height 1.6, weight 60 kg. He discovered fire. They discovered fire. They started to live in groups and they hunted together 19 lakh year ago. Homo neanderthalensis, height 1.65, weight 80 kg, and they were buried, means they actually buried those who died. Specialized tool they use. They are able to make specialized tool for hunting, say 150,000 years ago. Homo sapiens, height 1.7, 70 kg, and artistic work started to take place. There are specialized tools being made of bones and horns 120,000 years ago. These are the stages of human development.